Okay, good morning everybody. As I was told you, my name is Andreas Bremges, and I will talk a bit about the challenge data that we use in CAMI and the simulation pipeline that we engineered to actually produce all this challenge data. And I'd like to start with acknowledgement my co-workers without whom none of this would be presented today, which is Andre and Fritz, who's on the second row right there. I will show some of our toy benchmarking in the start as a motivational example. And Peter Hoffman, who was really spearheading the development and anything regarding to the pipeline. Okay, so big thanks to you guys. So I don't know if you've noticed, but CAMI was recently featured in an article in Nature Methods, and it was called like Microbiology, the Road to Strain Level Identification. And yeah, for very many medical studies, it's really important to tackle down or to identify the exact pathogen strain. And when we designed CAMI 2 years ago, we had this in mind already, because strains are important. And coming myself from an assembly background, I'd like to just show you one toy example that strains are not only important to identify known strains or pathogenic strains, but also have an impact on de novo assembly, de novo metagenome assembly. And this is something that I did with Adrian. And what you see here is we just constructed a minimal challenge example of 2 microbial genomes, one E. coli, I think it was, and one that we evolved using a strain evolver from Aaron Darling to have a different average nucleotide identity. So on the x-axis you see the average nucleotide identity between the 2 strains, and we use 2 recent and modern metagenome assemblers to assemble a mixture of those two strains. And what you see on the y-axis is the genome fraction. And let's look at the megahit ones at the top first. What you see is basically that megahit reconstructs pretty much all of the genome if the strains are fairly distant. Okay, they do not. They have an average nucleotide identity of at most 95%. But then you have this, I don't know, twilight zone or whatever you wanted to call it, where the assembly performance dramatically drops. And what you see here in the different with different alpha values is basically how you want to evaluate this. Let me first explain what happens. Okay, as the strains become more similar to each other, the assembly begins to break even more, and then at one point the strains are so similar to each other they get merged into one consensus. Okay, this happens around here, I'd say 99.5%. And what you measure here with is basically, well, we have some consensus assembled of our strains, but we cannot distinguish between the 2 strains. So if we were to benchmark it to say, well, I want both strains assembled, both copies, we only assemble like 50% of each strain because we collapse them. Was that clear, or should I iterate it once again? Let's do it again. Do it again. Okay. So let's start at the other half of 99.5% identical strains. Okay. We have a mixture of those strains, only those 2 strains. They're 99.5% identical. We want to assemble both of them. And look at the top. Megahit, what it produces is one genome, which is a consensus representation of both strains. So it collapsed both strains. Okay? And so for each strain, we have roughly 50% of the genome recovered. Okay? If we, this is, for most settings, it's perfectly fine to have like a consensus contig or consensus strain representation. But if our goal or our ultimate goal is to reconstruct the strains, the assembler failed. It couldn't do it. Okay? And these are the different evaluation metrics. Here we actually look at was each strain reconstructed, and here in the lighter colors we say, well, it's fine if it's a consensus thing. Okay, was this clearer? Okay, and well, the challenging part is here between 95 and say 98% average nucleotide identity between the 2 strains, because there it's too similar to get told apart by the assembler, but it's not similar enough to get collapsed into the consensus contig, and the assembly just breaks. And a similar thing we see with MetaSpace, which was just published some weeks ago. Again, it cannot reconstruct both strains once the average nucleotide density is below or above some critical threshold. And the interesting thing is they even claim that they focus on producing these consensus strains or consensus contigs, which is the lighter part here. Okay, so strains are not only an issue with profiling or binning, but also vastly impact the performance of de novo metagenome assembly. So enter CAMI. You've seen this before, and most of what I will be talking about actually happened in 2014 or was started in 2014 in our public workshops, the hackathons, and internal developer meetings, where we actually sat down and thought, well, how do we want, or what data do we want to give to the community? It should be really realistic and or as realistic as possible. And what came out of it by contributions by dozens of people, I'd say, and was spearheaded by Peter Hoffman later on, is basically what we call metagenome simulation pipeline. If you have some sexy name in your head, just drop us a hint. For now, it's just a working title. And basically, what we wanted to achieve, and I think what we really achieved, is to have one pipeline to go from your genomes, to annotate them, to simulate realistic data sets with different experimental settings. So whether it's just one snapshot of a metagenome sample, whether it's differential abundance or time series, this is everything baked in there. And it's like 99.5% Python. I think I checked in one Perl script just to screw the statistics here. And it's really modular, and you can extend it. And if you want to discuss it in detail, Peter is the right person to do that. And I will just walk you through the excellent documentation that I believe most of you haven't seen yet, because I'd like to highlight that everything is really extensively documented. So we have a design documentation living on GitHub. It's so far private, will be made public soon. And so the pipeline mainly consists of two parts. The first one is the genome annotation. The second part is the metagenome, the actual metagenome simulation. So we need to annotate genomes because for CAMI, especially for the profiling part, we wanted to have novel genomes in there. You can't just sample from the database of known genomes and give it to the community, and they use reference-based profiling or binning methods, and they will, of course, achieve brilliant results. But the reality is most metagenomes contain stuff that is unknown. And to come close to this goal, what we did for CAMI is asked our collaborators to donate genomes to hold back some genomes until the challenge is over. And so I will later talk or later show how many genomes and so we have. But with new genomes, we had to really introduce a rigorous QC to check what genomes can be part of the CAMI challenge to avoid contamination issues or misclassification issues. So classification was mostly done based on the 16S RNA gene. And so once we have a genome assembly from isolate genomes, we search for the 16S in there and then align it to the silver reference alignment of these 16S genes using capabilities of mother. And for that, then we get like the first taxon label for our known genomes, for our unknown genomes, sorry. Okay, so now we have for each of our CAMI genomes, we have the 16S, we have the first taxon label. And what we also do is to compare all the genomes against each other and the known genomes from NCBI, RefSec, JGI, whatever, to cluster them and organize them in OTUs to determine the novelty category. Okay, so whether this genome represents a new strain that has not been seen in a reference database before, or even a whole new family that we have no representative seen as of now, or even higher, but I think we do not have like new deep branching phyla, unfortunately. Okay, so we need this because we work with new genomes, with genes that were not available in the databases. And I'd like to use this spot now to actually thank our collaborators, in particular the JGI team, Tanya Wolke, Nigos Kapidis, and Eddie Rubin, who generated 311 genomes for CAMI. The MPI, who generated 446 genomes, which is Paul, Young, and Ruben. And the University of Copenhagen, especially Tour, who generated plasmids for us. So plasmids long have been neglected in all this benchmarking stuff, and we thought it might be a very good idea to mimic realistic metagenomes that, of course, contain plasmids as well, and spike some plasmids in there. So to sum up, we have almost 700 new genomes sequenced at the JGI and MPI, close to 600 plasmids available to sample from for our CAMI data sets. So the red ones here were just the portion that was included in our very strict QC step. For example, if in an assembled genome no 16S was found, we said, well, better be safe than sorry, throw it out. Or if there was like from the JGI, we got annotated genomes already. If our annotation was not 100%, did not 100% confirm their annotation, so we had like two different opinions, we said, well, throw it out. We definitely have enough genomes to sample from. Okay, any questions so far? All right. So what's next? It's the core step in the simulation pipeline, the actual simulation of our metagenomes. So, well, of course, it's a metagenome simulation pipeline. So what you get out afterwards, basically a bunch of FASTQ files generated from your input genomes. So the important thing is what happens, what is modeled in between. And so what's possible with the pipeline is to do first a grouping of genomes. So if you want to, for example, sample from different groups of genomes, and we did this in CAMI because we had one group which was the bacterial chromosomal DNA, and the other group was the plasmids. Okay, so we treat them independently from each other. We have some strains already sequenced, but we also simulated artificial strains using this SG Evolver by Aaron Darling, which is, comes with a morph package, which basically starts off with one genome and then uses some fancy algorithms to produce realistic or as realistic strains as possible. And we use this to enrich our data sets for more strain variation. And the last things is, well, we need to somehow select the genomes and sample from a realistic distribution. I would like to dive into that. So the genome selection itself, well, it's not completely random, but what we want to achieve is we want to sample or select genomes that cover most of the tree of life, so to say. So we do not want to just sample from one family, and therefore we take the novelty category into account when randomly selecting, say, 500 genomes or 200 genomes from our pool of 600 or 700 CAMI genomes. Okay, and also, as said before, we introduce artificial strains. to make it more realistic, more challenging for all tools out there. And the distribution, well, also quite motivated by realistic data sets. It's commonly expected that the abundance distribution follows a log normal distribution, and this is our underlying distribution where we sample the abundances from. And we have different modes that we can use the simulation pipeline in. We can just do one metagenomic snapshot of one sample, so we just simulate it once. We can do replicates, so draw twice from the same distribution. We can do a time series or a differential expression stuff, and I will show you on the real CAMI datasets how this actually works down. But first of all, we have 3 different datasets that we define as the CAMI challenge, the first CAMI challenge. And the first difference is that there's a different degree of, or different amount of genomes in there. So we call it here the low complexity one, which has like 40 genomes and 20 plasmids. The medium one, total of 220, 230 genomic elements, and the high one, which is really a high diversity one. And what you see here in green is the amount of plasmids in the samples or in the data sets. Dark blue are the real sequenced chromosomal DNA, and light blue is how many strains did we artificially simulate using Aaron's evolver. So looking at this, you might say, well, it's only a few strains, but the ground truth is it's really a lot of strains, because even on the sequenced genomes, we have a lot of strain variation covered. So dark red is basically overwhelming. Those are the strains in there. So by strain, heads up to Peter Bellman and Alex Scherber did this analysis. So basically what is shown here is for all genomes in the CAMI data sets, we did all the pairwise comparisons as outlined before, and check whether there is a second genome in the data set that has an average nucleotide identity of 95% or higher. And so we call this then, well, there's another strain in there. Okay? And so, for example, for the strain data set, you see that only like 200 of our 600 genomes that are in there are unique species without any interfering strain. And the other 400 are genomes that have at least one very similar strain in the same data set. David, you look like you have a question for that, or is it just... No, okay. Anyone else? Okay. So I personally think it's quite a challenging data set for assembly because you have lots of strains in there in this pit of 96 to 98% sequence identity that usually break your assembly. But also for profiling and binning, it might be a challenge if your aim is to actually go down to strain level, where it becomes interesting, but where it also becomes very, very hard. So this is the CAMI low data set, and this covers 6 different phyla, 20-something genomes, and what you see on the x-axis is the relative abundance on the log scale, basically this line that you would expect from drawing from a log normal distribution. Okay, the colors are the different phyla, just to show you that it's well branching into the six phyla in there. Basically, just as a reminder, yeah, it pretty much looks like you would expect it. Okay, the medium data set is higher; it's 70 or 80 genomes, and what we did there was we wanted to create a different data set mimicking this Albertson style experimental setup where you have like one metagenomic sample and you, for example, do different DNA extraction methods which highly skew your abundance profiles. So we started off with one distribution and then simulated the second sample totally independent from the first, and this is what we get from it. basically two log normal distributions independent. They are not really correlated. What you see here is now each point is one genome, relative abundance in the first sample, relative abundance in the second sample. So this should aid, for example, these reference-free binning techniques that require an assembly and then look at differential abundance patterns alongside with nucleotide composition. So this should really where those methods can excel. The last data set that we have is, would go like 2 or 3 meters in the ground, is the high complexity data set. Okay, 400 genomes, 200 plasmids. And what we did there is we want to simulate some time series experiment. So again, the initial distribution is drawn from a log normal distribution, and then we basically wiggle around these initial values, again drawing from log normal, and therefore produce a distribution of these genomes that are dependent on the previous states. Okay, and we do this for up to 5 time points in the CAMI high data set, and this is what it looks like in the end. Just scrolling down to the end, so again, relative abundance on the x-axis. Okay, any questions so for the CAMI data sets so far? Okay, I'm faster than expected then. Pretty much what we end up with is then we have 3 data sets. We have our abundance tables where for each genome we have, or for each strain, we have our relative abundance, and this we basically feed into the ART sequencing simulator. And big thanks to Wai-Chun Huang, and apologies if I pronounce it not correctly, who provided us with a realistic error profile derived from an actual high-seq run to mimic the Illumina sequencing errors and biases. And what we end up with is those 3 data sets. Below one sample, we choose the fragment size of 270 bases, which is like the standard Illumina protocol, the cheapest one that most people do. Sequence 2 simulated paired end data sets of 150 base per read length, and sequence it or simulated 15 gigabases. The medium data set, 2 samples, differential abundance setting, 2 different fragment sizes as well. So we have a long fragment or long insert here of 5 kb. Total sequencing depth or simulation depth is 40 gigabases. The highest one is the time series with 5 samples and the sequencing depth of 75 gigabases. And that was basically handed to the community to try their tools on, and I'm really looking forward to digging into the results with you these days. So thank you. If there are no questions, I would take hand over to Peter. But yeah, sure.
Will you go back to your second slide, I think?
Second slide, yeah.
Mini.
Yeah.
Mini method.
Yeah, sorry about the confusion in the beginning. It was just not warmed up yet. Yeah.
So, when you look at the consensus,
Yes.
you look at genes also? And what happens to genes? Are they all...
So your question is whether genes are truncated
Yeah,
by misassembly errors? No, we haven't looked at that in detail yet.
okay.
This would
Because
be,
the genome fraction, I mean, of course you want to look at the genome fraction first.
yeah, this is like the most,
next question would be
yeah.
what happens to the genes? Can you still predict genes correctly, and how do the genes look like?
yeah, this is definitely something interesting to look into. Once we reach like 99 point something average nucleotide identity, I would assume genes are not affected too much, because they're just very, very similar, and usually they are much more conserved on protein level than on nucleotide level. But yeah, we will look into it. Thank you.
I do have a question now. When you have the horizontal bar charts a little further down,
Which one's the first? No, I think this one is not really interesting, but the other one. This one?
Those guys, okay. So where does the novelty show up in these guys?
It's not in this bar chart.
Can you give some sort of indication, like?
So you mean how many novel families are in there, how many novel phyla are in our data sets? Yeah, I can dig it up and talk to you this afternoon then, or talk to you, if it's of general interest, to all of you and give you an update on that.
Yeah, I think that'd be interesting too.
Okay.
I looked up the high data set this morning. It was, I think, around 2/3 were classified as new species and 1/3 new genus and the high complexity one, and like a handful of new strains. The other two have also included new family and a few of new order ones in the low complexity one.
Only very few new families in the world.
I don't know how many new families.
Okay, well,
Because it'd be interesting to see, like, for the new order and the higher level taxa, what their relative abundances are, like in that sort of like log plot that you had up here, just to see.
Yep.
Okay. Yeah, I'll dig out the numbers and come back to you. Yeah, go.
Can we go back to the second slide again?
Second slide, strange stuff. Yeah.
Like is it so? Like they start diverging around 95, 6, 7. So is that not? I mean, it's a nice slide to like keep this in mind, but it's very natural, right? Because you have a little variation plus a little. Like if you have 100 nucleotide reads, then if you have there's a lot of stretches with 0 variation.
Yeah.
So that can never be overcome if you, as long as you have 100 nucleotide reads, right?
Yeah, I think yeah.
It's an interesting slide, but it's also a natural consequence of the reasoning.
Yes, it's no groundbreaking new insight. It's just what people long suspected and was gut feeling of many. It's just I personally haven't seen it in an assembly setting reduced to like the minimal constraints needed to make this point.
I don't know, I very much
Yeah.
like it, but it is also a natural. I mean, there's nothing in the assembler. Like, how could the assembler ever differentiate two things that are identical?
Well, it cannot be, that depends on the camera, not on the read length.
Yeah,
Well, depending on the
you can
assembler,
have
of
a
course. Right?
greater case.
Yeah, and so for metaspace, it actually uses well, I don't know how they call it, coupled or paired de Bruijn graphs. So they take the information from the whole fragment size, not only like one kmer, but still, yeah, what's not in the reads what cannot be explained by fragment size is basically lost. Yeah.
It really depends on if you keep the information about the read, right? And if you just do the camera de Bruijn graph kind of assembly, you're losing all the information about the reads, right? So even if the reads are like 5 kilobases long, it doesn't help.
No.
But of course, you could use this information.
Yeah.
If you would incorporate this into assembly, but usually they use it as
Yeah, but even in like an overlap layer consensus approach, you would have some person identity threshold where you just collapse things, and this I think would be around 98, 99% in most assemblers. So this probably also explains why, at least if you're just interested in consensus context, it goes up again. Also in these cases.
I think that the point would be that it's the same sequence in both strains. The assembler, you don't know that it's two strains there. Because it's one sequence, the assembler could maybe say, hey, it should be two strains for the same sequence.
Yeah, if the information is there in the data that you put into it. But
Yeah.
in this case, there will be a lot of stretches where it can't see any difference.
Yeah,
One
the
folder.
sequence is identical, but you could potentially decide it should be different. That it's not from one genome.
Yeah. And so I think the point is that most assemblers are pretty greedy collapsing strains, and this is something that I would question actually, and I would appreciate if the assembly community would move, as they do already, into these graph-based representations of assemblies, and probably just output the graph then, and you can then traverse the graph and see, well, we have some strain mixture in there. But yeah, we'll see.
Are there candidate phyla among the genomes for the
As far as I recall, unfortunately not. This was like, of course, this would be like a real treasure, but this data is really hard to get into our hands to use or to spit out there and challenge most people. If they have new candidate phyla, you would expect really interesting novel biology in there, and I don't know if I would donate it to CAMI to do that.
We had some authors, I think,
Yeah.
I think it was speaking to some people who had data from really deep branches. But the thing is, they usually were derived using some binning method. So then you would have some kind of circular logic. So the binning method was so we don't have cultured isolates from novel phyla, which would give us some unbiased standard of truth, and that's why we thought, well, maybe not. If we get single-cell sequencing data to be of that quality that it could be considered as a pure genome, that might be an approach.
I just looked it up. We have one new order.
Oh,
That's the best.
I don't know about the single-cell sequencing. Alex, you know more about that, right? We discussed using single-cell sequencing data to provide.
Yeah, we did. I don't know why we. I think it's just hard to get the data, right? Because people don't. I don't know. I
There was
don't remember
quality
actually.
issues also, wasn't it? That you sometimes have mixtures of different, so it's not really.
Oh, yeah, that's the other problem. Yeah, it's pretty hard to get clean samples, and we have to be certain that the samples are clean. Actually, if we include it in the challenge, so I think that's why we didn't use it.
I mean, my thought it's possible to get data.
Probably, yeah.
Genomes you have, are they complete or were they assembled?
So they originate from pure cultured isolates, and they were mostly sequenced on Illumina, the high-seq, so we have draft or extended draft quality status. They are not closed, if that's what you're asking for. No. And we simulated our reads from those draft genomes then. So it's dozens to low 3-digit numbers of contigs.
Continue from his question. So the criteria, as I can tell from the slide, is that there need to be an intact 16S in the genome, or otherwise you will throw the genome away. Is that right?
This was one quality measurement. Yeah, if we have like a confirming 16S, yes.
Yeah. Well, and we got the genomes early on, right? The study the MPI was doing. We got the data when they were still assembling the genomes, and they didn't actually know what kind of genomes they had, right? And at that point, we already got the data, and we assembled all the genomes ourselves. And then some of the genomes seem to be contaminated. But contaminated by, it's probably not a clean like culture, but it had like 2 genomes inside. So these kinds of cases we just excluded, just to be sure that it's just one genome.
Wait, you did this based on what?
Basically based on clustering, yes, on tetranucleotide frequencies. So very simple. Whenever 2 clusters came up, we just discarded it.
I looked up if there were like 2 16S genes in one. This was already suspicious, and it was a subset of those Alex also thought they are suspicious, so we threw them all out.
Yeah, so what we also did was this GC, plotting GC with this coverage, and when you see like 2 peaks in there, it's pretty clear sign that the sample is contaminated. This is also what Alex was doing. So it involved a lot of manual curation also. It's not fully automated.
Well, and then you didn't talk about why we actually simulated it, because we had all the raw data, right?
Yeah, sure.
We could have used the raw data.
Yeah, do you want to explain, or should I take over? Okay, so the point is, what we do in the CAMI challenge afterwards is we want to have some ground truth to evaluate against. Sure, we could use the real data, but it's much easier and probably also cleaner to work with simulated data in the first challenge, because for each read we know where, from which genome it was simulated, it was sampled. We know the exact location, we know the genome. So for profiling and binning, this makes it fairly straightforward. Assembly also, because we know which regions of our assemblies were covered by simulated reads and can be reconstructed. And this is what we call a perfect assembly, and I think Alex will talk about it in his assembly evaluation stuff a bit more.
Real data, do you mean that you're trying to create some mock communities, maybe say in the second or third challenge later on? So I understand that you're trying to do some simulation for the first challenge, but when you're talking about real, are you trying to create some mock by throwing every cell together?
So this would be something to discuss, because there are just so many factors playing in, like you cannot measure the exact amount of input DNA and stuff like that. When I talked about real data just now, I meant, well, we have the real sequencing reads, why don't we throw these at the challenge data set? And instead simulated reads using the art simulator. But yeah, it's just to ensure like an easy benchmarking and evaluation afterwards, and also because they were sequenced at different centers. And you could probably also tell by, I don't know, the QC distribution of the reads, which center it came from, which helped binning or something like that. So just to make it all comparable. But this is definitely a direction that we want to move into.
There wasn't any mention of viruses. Were there viruses included in how much?
You mean the plasmids, or
It's not pure plasmids.
Okay.
It's not.
Okay.
Okay, so like by plasmids, you mean like plasmids and viruses? And
Yeah, it should actually be circular elements.
yeah. Okay.
Okay. Sorry. Yeah.
I mean, I would very much like to take 2 minutes at some point to explain the data set.
Yeah, so excellent. Would you want to do it right now? Or?
Yeah, sure.
Yeah.
So this
I'd love
data
to.
is different from all the data in that it doesn't come from pure cultures, it comes from metagenomes. So it has already gone through this pipeline that everybody's using for it. So it's relatively easy to assemble. It has high diversity, so it can actually be, like this is like a perfect situation where you can actually assemble it. So you just need to do it and notice that you have done it. So that's basically it. So it comes from metagenomes from rats, and they are pretty small, and there's both single-stranded viruses probably, and double-stranded plasmids and whatnot. So basically, they are not, like the assembly part of it is not so much an issue for them, else is the identification that what you have is a plasmid. That what you have is actually complete circular structure. And in my mind, people are doing that way too little in any assembly, because we're getting close to getting not just full plasmids, but also full genomes.
Thank you.
Okay, that means so the data, so again, the data comes from like metagenomes, so there were metagenomes sequenced,
Yes, but
sampled, and then you identify the
yeah, so
plasmids.
it's not really it's metagenomes where we remove all non-plasmid, non-circular DNA.
Oh, okay.
With the experimental technique, right?
Yeah, with
Okay.
experiments.
And that's before sequencing.
Before sequencing, yes.
So again,
And we know, I mean, we can see that there's no 16s. There's no stuff that looks like 2/3 of all, no, 1/3 of all contigs are circular of what we get out. So we know that this is pure plasmid stuff. Or circular element stuff. But so one big difference from real life plasmids is that real life big plasmids, they would be littered with repetitive elements. And these are not, because they can actually be assembled. And that way it does not represent a true life situation. But then again, those repetitive elements you would also find everywhere in genomes. So I mean, I think you have that part covered by the genome.
Actually, we should explain how we simulated these, because these were part of the simulation pipeline also. And of course, you cannot, your sequence is not circular, right? If you have a FASTA file, it's not circular. And when you run the sequence simulator on it, it will never simulate a read that spans the ends, right? And that's why what did we do? We made like 2, 3, or 4 copies of the plasmids. 5? 6.
At least 5. I remember 5.
So we made multiple copies and then just concatenated the sequences, right? And then simulated the reads, and then of course adjusted the abundance and so on. So that's how you could actually assemble the circular plasmids out of the data.
So what is the sizes of the plasmids? Say in the maximum or average? Do you have any idea?
They're relatively small because what you get out, so the experimental technique will remove anything that's linear, and most big plasmids will break somehow in the purification process. So what we have, the biggest I have assembled with this technique is a phage actually of 33 kb, and that's not included here. So I would say that the biggest is probably around 15, 20 kb, and then down to 1,000 nucleotides.
Okay, well, if no more questions, I have to thank Andrew again.